Hello students, welcome on PARAVET digital platform. In last class, we have discussed about introduction to cell. In this class, we are studying cell organelles. Before it, we take some revision of the last class. At first, Robert Hooke discovered cell. Cell which was a dead cell, only cell wall was there because he took on his experiment the cork. First living cell discovered by Leeuwenhoek, which was protozoa, bacteria, and spermatozoa. And some discoveries of microscopes like Z. Jensen and H. Jensen discovered compound microscope. E. Knoll and Ruska discovered electron microscope. Resolving power of electron microscope 5 angstrom. Largest cell of animal kingdom ostrich egg, smallest cell PPLO, and longest cell nerve cell. Robert Hooke experiments were published in Micrographia book, and father of modern cytology is CP Swanson. Let's discuss today's class. We are discussing cell organelles, which are the parts of cell, or cell is made up of cell organelles. The first cell organelle is cell membrane. Cell membrane is the outer covering of cell which protects the cell and adjoins other cells surrounding. Cell membrane is present both of the cells, plant cell and animal cells. In plant cells, there are a extra layer, extra covering of cell wall around the cell membrane. Means here, assume this is plant cell. In plant cell there is cell wall, the outer layer of plant cell, and in animal cell there is only cell membrane. Cell membrane is also present in plant cell as well as animal cell. This is animal cell. So in veterinary we are talking about animal cell. The outermost covering of cell is cell membrane. The outermost covering of a animal cell, and it is made up of phospholipid and protein. Cell membrane is made up of phospholipid and protein. Means type of lipid having phosphorus moiety called phospholipid with proteins. Cell membrane width means thickness. Approximately 75 angstrom. We know 1 angstrom is equal to 10 raised to power minus 10 meters. This is history of plasma membrane. Cell membrane is also called plasma lemma and plasma membrane. There are 3 models which are popular. First is, for exam point of view, unilayer model or single layer model was given by Robertson. Tri-layer model, or we can also say uh, sandwich model, which was given by Danieli and Dawson. And the third model, which is most widely popular, Singer and Nicholson model, fluid mosaic model, which was given by Singer and Nicholson in 1972. In exam, it is very important in all the models, and we will also discuss about fluid mosaic model. The phospholipid layer is bilayer. Phospholipid is bilayered, and the three models of cell membrane, we are talking about only important models. First is single layer model, which was given by Robertson. Second model, tri-layer model, and also known as sandwich model, given by Danieli and Dawson. And third, most widely popular, fluid mosaic model, given by Singer and Nicholson in 1972. Three models of cell membrane. Cell membrane is also called as plasma lemma. Plasma lemma or plasma membrane or cytoplasmic membrane. There are various names of cell membrane. Means plasma lemma is the old name or classic name of cell membrane. Now we are discussing functions of cell membrane. Cell membrane functions, first is to protect. Functions. First is protection. Protection. Because of outer covering, cell membrane controls inside and outside molecules flow. Means it controls or regulates cells internal environment by endocytosis and exocytosis. Phagocytosis. Phagocytosis. Phagocytosis is a type of endocytosis. Endocytosis means taking a material or molecule inside the cell. Taking inside the cell. And the another term is exocytosis. Exo means exit. This is cell, and the waste materials and excretory products from cell are exocytosed by cell membranes. It is exocytosis, it is endocytosis. And endocytosis is further divided into phagocytosis and pinocytosis. Pinocytosis. Phagocytosis means, phago means taking a solid material by the cell membrane inside, and pinocytosis, pino means drinking, taking a liquid material inside the cell. Like this is cell and this is fluid portion. Cell membrane takes fluid portion, fluid material inside the cell. It is called pinocytosis, drinking. Pino means drinking. Phagocytosis, this is cell when it takes any solid material, it is called as phagocytosis. Solid, and there is liquid. When liquid material or liquid food or liquid molecules taking by the cell, it is called pinocytosis. Phagocytosis, phago means phagocyte means eat, and the solid particles are eat or taking by the cell membrane inside, then it is called phagocytosis. Next work or next function of cell membrane is formation of microvilli. Microvilli are the finger like structures which increases surface area of cell membrane. Like this, microvilli. Microvilli means they increases surface area. Next function of cell membrane is selectively permeable. Selective permeability. Selective permeability means selective materials are taken and selective materials are given up from the cell membrane. It is selective permeability. Next, active and passive transport. When a molecule is taken by consuming or by wasting energy, then it is called active transport, and according to uh, gradient of concentration, when a molecule goes, then it is called passive transport, means there is no energy consumption in passive transport. There is any energy consumption in active transport. So these are the some functions of cell membrane. Now we are studying the structure of cell membrane. This is cell membrane, and the most widely popular, the most famous and most acceptable model of cell membrane is fluid mosaic model. Fluid mosaic model given by Singer and Nicholson in 1972. According to this model, there is bilayered phospholipid layer, and This type of phospholipid molecules, the head and tail, the head and tail. Head is hydrophilic. Hydrophilic means it have tendency to bind water, water molecules. And the tail portion, this is head, this is tail portion, it it is hydrophobic in nature. Means it repels and repels from water, not interacts from water. The bilayered phospholipid, in between there are various molecules of proteins. Like these proteins are channel proteins which helps to inside and outside, means excretion of molecules from cell. Some carbohydrate chains are present which are important for identification of cells, like antigen antibody reactions. These carbohydrate chains are important for recognition of a cell. Proteins are of two types: intrinsic protein and extrinsic protein. Outer surface there are extrinsic protein, and inner surface intrinsic proteins or integral proteins. The most widely accepted model of plasma membrane structure is fluid mosaic model of Singer and Nicholson 1972. According to this model, cell membrane is composed of a fluid lipid bilayer with a consistence to changing mosaic pattern of associated proteins. These bilayer phospholipid layer, and there are various proteins inside the cell: integral protein or intrinsic protein, outside the cell extrinsic proteins, and these proteins helps for the transport of molecules inside and outside. This is fluid mosaic model, and in other words we can say the fluid mosaic model in which phospholipid in which phospholipid is in the form of semi liquid, and protein molecules floats on this semi liquid phospholipid like. This is comparison and important comparison, like iceberg in ocean. Like iceberg in ocean. Icebergs floats in ocean this type. So it is structure of cell membrane. Next cell organelle is endoplasmic reticulum. At first we are talking about the scientist who discovered. In the last organelle cell membrane discovery was cell membrane discovered by Carl Nageli. And now next cell organelle is endoplasmic reticulum. The endoplasmic reticulum, endo means inside, plasm means life or living matter, endoplasm means living matter. Reticulum means a network like structure or a mesh like structure. In the cell, from the nuclear membrane to cell membrane, there is a network of tubules, vesicles, or a irregular network of tubules which is called as endoplasmic reticulum, discovered and name also given by Potter. Discovered by Potter. Endoplasmic reticulum is the cell organelle which is distributed from nucleus to cell membrane. The literal meaning of endoplasm is living matter. Endo means inside the cell. Means as cytoplasm or protoplasm, which is another words we are saying endoplasm. Reticulum. Reticulum means mesh-like structure, mesh-like or network. Reticulum means net-like structure or mesh-like structure. Discovered by Potter. It is the irregular network of. It is the irregular, as in the picture we are observing irregular patterns of tubules, and the tubules are unimembranous, means single membrane is there. It is the irregular network of single membrane tubules which is distributed from nuclear membrane to cell membrane or run out from nuclear membrane to cell membrane. Distributed from nuclear membrane to cell membrane. Means inside the cell there is a network of irregular tubules which is called endoplasmic reticulum. Now important points from endoplasmic reticulum exam point of view. On the basis of exam we should remember these points. First is it forms or it makes internal skeleton. Internal skeleton of a cell. Like bones makes our skeleton, like endoplasmic reticulum makes internal skeleton of a cell. Second, it is also called endoplasmic reticulum, we can say in short form ER. ER is also called highway of cell. Why it is called highway of cell? Because it transports fat and protein molecules through it. As a highway for vehicles are transported from highway. Likewise, endoplasmic reticulum helps or it facilitates transport of fat and protein molecules. Endoplasmic reticulum ER is also called as highway of cell because It transports fat and protein over it. Endoplasmic reticulum is important for the formation of Golgi body. Means Golgi body generates from endoplasmic reticulum. And Golgi body forms lysosomes. It is a serial, remember it, important. Endoplasmic reticulum generates or forms Golgi body. And Golgi body makes or forms or generates lysosomes. It is a important formation order because one cell organelle is made up of other or is forming by other cell organelles. Important is Golgi body forms by Golgi bodies formation occurs by ER. Endoplasmic reticulum makes Golgi bodies. Next point, which is also important, it gives the place for synthesis of ATP in the cell. It gives space for synthesis of adenosine triphosphate molecules, ATP. ATP synthesis on endoplasmic reticulum. Now we are classifying ER. Endoplasmic reticulum is of two types, RER and SER. RER, the full form is rough endoplasmic reticulum. SER means smooth endoplasmic reticulum. Why it is so called RER? Rough endoplasmic reticulum. Smooth endoplasmic reticulum. Because there is presence of ribosomes. We know ribosomes are the smallest cell organelles inside the cell, and when they attaches any cell organelle, they are like granules, like this. This type of endoplasmic reticulum, on its surface there are ribosomes in granular form. Ribosome is made up of two units. One is small unit, subunit, and the another is large subunit. The large subunit is attached by ribofferrin protein to endoplasmic reticulum. This is like ribosome structure, and ribofferrin protein is there. It helps. This is ER. So ER and ribosomes. Ribofferrin protein helps of attachment large subunit of ribosome to endoplasmic reticulum. Because of the main function of ribosome is protein synthesis, so the main function of rough endoplasmic reticulum is to synthesize the proteins. Because there are proteins, there are ribosomes which synthesize proteins, so this RER or this endoplasmic reticulum synthesize proteins. Means main function of RER to synthesis of protein. The another type of endoplasmic reticulum is smooth endoplasmic reticulum. Because of smooth, why it is smooth? Because there are no ribosomes on its surface. Like this, like this. The smooth surface is there, but functions are important for exam point of view. Smooth endoplasmic reticulum synthesize cholesterol, synthesize steroids, synthesize hormones, synthesize vitamin C. Smooth endoplasmic reticulum and also detoxification. Functions are detoxification, synthesize steroids, synthesize cholesterol, synthesize vitamin C. These are the functions. Various hormones are made up of steroids and cholesterols. So we can also say hormones are also synthesized by smooth endoplasmic reticulum. Now there are three components of ER. There are three components of ER. Endoplasmic reticulum is composed of three types of components. First is cisternae, second is vesicle, and third is tubules. Cisternae are the closed parallel structures. This type of structure, long and flat structures which are arranged parallel to each other. Parallel to each other. Second, vesicles. Vesicles are sac-like structures which may be oval, elliptical, and other shapes. Oval, elliptical, sac-like. And tubules are this type branched structures. And the remaining we can clear from this diagram. This is nucleus. Around the nuclear membrane there is endoplasmic reticulum, and the red beads we are seeing in this diagram, which are ribosomes, which we are presenting. Ribosomes, where the presence of ribosomes, this endoplasmic reticulum is rough endoplasmic reticulum, and there is absence of ribosomes, which is smooth endoplasmic reticulum, SER, smooth endoplasmic reticulum. Because of formation of Golgi apparatus by endoplasmic reticulum, in Golgi apparatus also the structure is same to like endoplasmic reticulum. So we have studied the endoplasmic reticulum. Now endoplasmic reticulum is absent in RBC. Mammalian RBC is somewhat different structure than a simple cell or a simple animal cell. Absence of endoplasmic reticulum in RBC. In prokaryotic cell, there is also absence of endoplasmic reticulum, and in the muscles this endoplasmic reticulum is called sarcoplasmic reticulum, which provides calcium ions at the time of contraction of muscle. In muscles, endoplasmic reticulum, is called sarcoplasmic reticulum. Sarcoplasmic reticulum. Because the study of muscles called myology, the another name of myology is sarcology. The word sarco is provided for muscles. So, sarcoplasm is the internal cytoplasm of a muscle fiber, means sarcomere. Sarcomere is the unit of striated muscles. Now we are talking about sarcoplasmic reticulum, which is the another name of endoplasmic reticulum within the muscles. And sarcoplasmic reticulum provides calcium ion for the contraction of muscles. So we have studied two cell organelles. One is cell membrane and another is endoplasmic reticulum. Thanks.